Welcome back, everyone. So we are right in topic 4. So we're going to continue on with the plasma membrane this week. We're going to take a closer look at how that plasma membrane is semi-permeable. So it allows certain things into the cell and out of the cell. So this is chapter 4 in our OpenStax textbook. If you're using the Campbell Biology textbook, it would be chapter 5. So this week we're going to look at 3 sections from our textbook. So the first section we're going to look at passive transport with this video, okay? And then the next section we'll piggyback off of that with active transport. And then at the end of the chapter we're going to begin looking at the interior of the cell. So basically how the cell obtains energy and take a look at metabolism, which is basically just all the chemical reactions that occur inside the cell. Okay. So in this particular section, 4.1, we're focusing in again on passive transport. So we'll learn about how things can enter the cell through passive transport. So that's basically just the most direct form of membrane transport. And it's something that just naturally occurs, that doesn't require any energy to accomplish. It's just where substances move from high concentration into areas of lower concentration through something called diffusion. So we'll talk more about that in this video. So by the end of this video, you will be able to explain why and how passive transport occurs. You should also be able to understand the processes of osmosis and diffusion, as well as define tonicity and describe its relevance to passive transport. Okay, so we'll get into understanding like concentration gradients and what that has to do with tonicity. Okay. So let's start out with the what. What is passive transport? So in the last week we learned about the plasma membrane being selectively permeable. Okay, so because of that phospholipid bilayer, only certain things can pass through it. Imagine that this is kind of a simplified view of our plasma membrane, and I gave that analogy of our plasma membrane kind of being like a gate, okay? So, or maybe even a fence, okay? So certain things can squeeze through, but not everything, okay? So some substances can make their way through, but not everything. So the ones that can pass through, they can pass through the membrane through passive transport. So passive transport, it doesn't require any energy. It can just pass, these substances can move through very easily. And the way they do that is through something called diffusion. So what is diffusion? So diffusion is the ability for substances to move from an area of high concentration to an area of low concentration. Okay, so imagine you have an air freshener, okay, and you spray that air freshener. So when you spray that air freshener, initially the odor particles are at high concentration, right? Okay, and then slowly they disperse through the air, okay, and so as they disperse through the air, the freshener odor particles move from that corner where you sprayed it all throughout the room. Okay, so they're moving from that high concentration to low concentration, and eventually you can smell the air freshener all the way across to the other side of the room. So that's what diffusion is. Okay, so imagine those little odor particles in that air freshener moving from that high concentration where it was sprayed and then over to the lower concentration, the other side of the room. Okay, so substances, they have that tendency to move from areas of high concentration to areas of low concentration. Okay, so that's what diffusion is. And they go with their concentration gradient. So a concentration gradient is a physical space with a different concentration of a single substance. So when we we'll talk about going with the concentration gradient. So that's going from high concentration to low concentration. Okay, so that's simple diffusion. We'll also talk about going against your concentration gradient. So that requires energy. So if you're going against your concentration gradient, you're going from lower concentration to higher concentration. So that would be unusual. So that would be like the odor particles moving towards the higher concentration. Okay, so that's unusual. So usually with our concentration gradients, they're going from high to low. All right. So this is something that we'll be dealing with as well in lab. So, you know, hopefully you'll get some good visuals with our videos and things like that. Okay. So passive transport requires no energy. It's just that tendency for substances that, you know, to want to move from high concentration to lower concentration, to spread out. Okay. So another analogy would be like if you could do this experiment at home where you just take like a glass of water and then just put like one droplet of like food coloring. So that concentrated droplet of food coloring, at first it's very concentrated. You see the actual color, that big drop of color. But then if you come back a couple hours later, the coloration has dispersed, right? Maybe overnight it's no longer, concentrated. It's all dispersed, and the whole glass of water is, slightly colored throughout. So that's through diffusion. So that coloration has moved from high concentration of the dye to low concentration. And so it's completely spread out until it's at equal concentration. Okay, so that's simple diffusion. Okay. So just to review, so our plasma membrane is selectively permeable, and it's selectively permeable because of those phospholipids. So we have that phospholipid bilayer. And if you recall, the phospholipid bilayer is amphithetic, so that means that it has hydrophilic heads. So remember, the hydrophilic part is water loving. So hydro means water, philic means loving, and the heads are the phosphate groups, so that's the polar group. And then the hydrophobic tails, hydro means water, phobic means spherines, and then the tails are the fatty acids. Okay, so that's the non-polar part. And then remember, it's a bilayer, so the hydrophobic tails of the two layers are facing each other. So we have this non-polar section of our semi-permeable membrane, and then the heads are facing outward. Okay, so because of the configuration of our plasma membrane, it's, this nonpolar region makes it so that semi-permeable. So only certain things can pass through simple diffusion, or passive transport. So lipid-soluble materials can pass through. So, lipid-soluble would be, nonpolar substances, so such as like vitamins, A, D, E, and K. So those are lipid-based lipid-based vitamins. So vitamin A, D, E, and K. Really small molecules with no charge. Okay, so like oxygen gas, O2, and carbon dioxide gas, CO2, those are very, very small molecules. They can easily diffuse across the plasma membrane. Okay, water can actually diffuse across as well because it's really small. Okay, and then some small ions can pass through. So if it's small enough to squeeze through the phospholipids, they can, pass through. Okay, and then things that are lipid based, they can pass through basically. All right, so those are things that can pass through the selectively permeable membrane. All right, so we're going to start off with an Amoeba Sisters video. And this Amoeba Sisters video is going to talk about diffusion. So give us an understanding of what diffusion is, and then we'll go into some detail from the book. Okay.
heard of the ick? The disease, that is, that can afflict aquarium fish? I will never forget the first time my fish caught ick. Well, it's called ick, it stands for this. So, ick it is. It's actually caused by a protist, a parasitic one. In our ecology video, we talk about parasites and how parasites benefit while causing harm to their host. And in my case, it was all of my beloved fish. Well, okay, technically it affected Gertrude first, but then it spread because it is a contagious disease in fish. Ick typically causes these white dots. It gets on their fins, on their bodies, on their gills, all over. It can even be lethal if not treated. So it's bad news, and being a guppy enthusiast, I was really worried about all of my guppies. This is not a video about parasites. This is a video about diffusion. So you're probably wondering, how is she going to connect diffusion to this parasitic protist disease? Well, it's because of the treatment. You see, there is a commonly purchased treatment for the disease of ick. One common treatment option contains an anti-parasitic called methylene blue. Its name is not misleading. It is quite blue. The directions on the back of the medication outlined how many drops to add per gallons of water. And I watched and tried to encourage my fish to swim towards the medicine. But they were panicked, maybe because a portion of their water was changing colors, but also probably because of the kid trying to get their attention to swim towards the medicine. But you know what I didn't know then? I didn't need them to swim over there. The blue drops would spread on their own in a process known as diffusion. Diffusion is when the net movement of a substance travels down its concentration gradient. That means it moves from a high concentration to a low concentration. When the methylene blue drops were added into the water, they didn't just sit there. The molecules traveled in the water through diffusion. The high concentration of molecules, which is where they were dropped, spread to areas of low concentration in the water. Pretty soon, the water had a uniformly blue appearance as the molecules eventually are evenly dispersed. This was very important for treating my fish because the treatment reached all of them, and they soon got better. Diffusion doesn't just happen in water. For example, it occurs if you spray an air freshener in the air. The net movement of molecules travels from an area of high concentration of the air freshener to a low concentration of the air freshener. Pretty soon, it's likely others would smell the air freshener, even if they were quite a distance away. Now, I want to mention two important points. First, we say net movement because that's the overall movement. But it doesn't mean that the molecules can't move around the other direction, and it doesn't mean that the molecules eventually stop moving either. The molecules are continuously moving, even when equilibrium is reached. It's just the overall movement, the net movement, is from a high concentration to a low concentration, until equilibrium is reached. Second, diffusion is a passive transport. That means an input of energy is not needed. We say that passive transport does not require added energy. When we talk about diffusion of molecules in cells, for example, the diffusion of oxygen molecules into a cell, it is classified as passive transport. A concentration gradient is itself a form of potential energy. Passive transport is different from other processes that we detail in our cell transport video, like active transport, where an input of energy is required. And speaking of our cell transport video, we outline that there is also a type of diffusion called facilitated diffusion. Facilitated diffusion is when molecules still have a net movement of a high concentration to a low concentration, but they may be too large or have other characteristics that prevent them from directly traveling across the selective cell membrane. Therefore, they have to go through a protein channel. It's still diffusion because it's still passive transport and it's still traveling down its concentration gradient. It's just that, in that case, it generally has to go through a protein to get inside. Now, there are factors that can affect the rate of diffusion. We'll mention just a few of them when considering these small molecules here, diffusing in simple diffusion. Distance. The greater the distance that needs to be traveled, the slower the diffusion rate. For example, you could observe a difference in a 5-gallon tank versus a 55-gallon one. Temperature. Okay, really quick, would you think a higher temperature or a lower temperature would increase the diffusion rate, assuming everything else is constant for the point of the comparison? Generally, a higher temperature. A higher temperature generally would mean there is more movement of the molecules, so the diffusion rate is increased. This has to do with energy, so check out the link in the description for more about that. Characteristics of the solvent. For example, is the solvent very dense? That could slow the molecules down and decrease the diffusion rate. Characteristics of the molecules, or whatever substance is traveling through diffusion, since technically not everything that travels in diffusion is defined as a molecule. What is the mass of the substance traveling? Generally, a substance with greater mass will have a lower diffusion rate when compared to a substance with less mass. Characteristics of the barrier, if diffusion is crossing a type of barrier. To explain that, let's consider diffusion across a cell, where the barrier would be the cell membrane. Small, nonpolar substances pass through a cell membrane easier than something large or polar. That will affect the rate of diffusion. The surface area and thickness of the cell membrane also will alter the diffusion rate. For example, a large surface area and thin membrane would generally allow a faster diffusion rate than a smaller surface area and thick membrane. By the way, there are more factors that can affect the diffusion rate than just those 5 we listed. In fact, you may have already thought of another factor when seeing our simple diffusion image. Generally, if I were to increase the concentration of the substance here, that would cause a larger difference between these concentrations, and that would also increase the rate of diffusion. Before we end, you might wonder, why should I care about this diffusion thing? Okay, other than the aquarium fish that I love, realize that diffusion is critical for all of life. Cells are the living unit that builds up all life, and materials that cells need to survive have to get in, and waste molecules of the cell have to get out. Now, while diffusion is certainly not the only kind of cell transport, diffusion is responsible for many critical items. One example? As you sit here right now, diffusion is responsible for oxygen leaving the alveoli of your lungs to your blood. Likewise, the waste gas carbon dioxide is able to exit your blood to the alveoli, a critical process made possible by diffusion. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so again, that was a good video that introduced us to diffusion, as well as gave us some information about what can affect the rate of diffusion. So we'll go through all of that in the next few slides. So again, diffusion is a passive process of transport. So it's that idea that substances move from an area of high concentration to an area of low concentration. And they'll do this until the concentration is equal. And like the video stated, once it reaches equal, that doesn't mean that the molecules no longer move across the membrane. They continue to move across the membrane just at equal rates. Okay, so you can see here that here we have a phospholipid bilayer, and on one side of the bilayer we have a high concentration of a substance. On the other side we have very low, there's nothing here. Okay, so this substance is going to move from high to low. Okay, so you can see it moving from high concentration to low concentration. And now it's equal concentration, but that doesn't mean that it stops moving. Okay, that doesn't mean that these molecules are now going to stay on this side and these molecules will stay on that side. They'll continue to move across the membrane, but just at equal rates. Okay, so this one might move here and this one might move there, but they'll move at equal rates. There will no longer be a net movement towards the cytoplasm, if that makes sense. Okay. And this is simple diffusion, passive transport, so it expends no energy. So this is just what we call kinetic energy. We'll talk about that in the third video this week. So it expends no ATP, okay, no cellular energy, all right? So movement of substances from high concentration to lower concentration until the concentration is equal. All right. And then the video discussed that diffusion can, the rate of diffusion can be affected by several different factors, and then the video described those factors. So one could be the extent of the concentration gradient. So if we have a greater difference in concentration, there'll be a more rapid rate of diffusion. Okay, so if we have basically a higher concentration on one side and nothing on the other, then it'll be a faster rate of diffusion. Okay, if we're already at basically, mostly equal, then it'll be a slower rate of concentration or rate of diffusion. So it'll be the extent of the concentration gradient will affect the rate of diffusion. Okay, so the closer the distribution of material it gets to equilibrium, the slower the diffusion rate becomes. Another factor that can affect the rate of diffusion is the mass of the molecule. Okay, so think about, the lighter the molecule, the faster it'll go. Okay, the heavier the molecule, the slower it will go. So definitely weight can cause the rate of diffusion to slow down. Okay. Temperature can affect the rate of diffusion. So remember, temperature can affect how the speed of these molecules. So if we have a higher temperature, we have increase in energy, so the molecules will be moving faster. And if they're moving faster, then they're going to be moving across the membrane faster. Okay. If it's colder, then there's less energy, and so the molecules will be moving slower, so the rate of diffusion will be slower. Right. And then finally we have solvent density. So solvent density can affect the rate of diffusion. So imagine if we have a more dense solvent to get through, it can slow down the rate of diffusion. So if we're moving the substance through water, it's going to be a lot easier for those molecules to move than, say, if we're moving through honey, okay, because honey is a lot thicker, okay, or Jello. We saw that image from the Amoeba Sisters, so it's harder for the molecules to move through, something that's semi-solid compared to something that's liquid. Okay. So those are things that can affect the rate of diffusion. All right, so we're going to watch a video on that. This is from your textbook, by the way.
Diffusion can also occur through cell membranes. However, cell membranes are selectively permeable. This means that some substances can diffuse through a cell membrane while others cannot. The flow of ions or molecules through a cell membrane as a result of diffusion is referred to as passive transport. It is referred to as passive transport as it requires no energy from the cell in order to carry a substance across the membrane. In the process of passive transport, the membrane acts as a filter. Phospholipids and protein channels control which molecules can cross the membrane and may influence the rate and timing of the movement of ions and molecules across the membrane. But they do not influence the direction of movement. Substances continue to move only from areas of high concentration to areas of low concentration. Let's now look at three types of passive transport that occur as a result of diffusion: simple diffusion, facilitated diffusion, and osmosis. Simple diffusion is the process by which gases, such as oxygen and carbon dioxide, dissolve in water, and phospholipid-soluble molecules, such as ethyl alcohol and vitamin A, diffuse across the phospholipid bilayer. This is referred to as simple diffusion because these substances can cross anywhere along the phospholipid bilayer, while most other types of diffusion such as facilitated diffusion, which we will look at in a moment, requires a substance to cross the membrane through a protein channel, or with the help of a carrier protein. Generally, the rate of simple diffusion of a substance across a membrane is a function of the relative concentration of the substance on either side of the membrane, the size of the substance's molecules, and on that substance's lipid solubility. In the process of facilitated diffusion, either channel proteins or carrier proteins facilitate the crossing of a cellular membrane by water-soluble substances such as sodium, potassium, and calcium ions, amino acids, and monosaccharides, which cannot typically diffuse through the phospholipid bilayer. As we saw earlier, each type of channel protein is typically lined with specific amino acids and has a unique diameter in order to allow only certain ions or other substances to pass. Channel proteins also often have gates which open and close in response to electrical or chemical signals. Carrier proteins, as we saw earlier, attach to specific molecules in the cytoplasm or extracellular fluid. The attachment triggers changes in the shape of the carrier protein that allows the molecule to pass through the protein and thus cross the plasma membrane. Some carrier proteins don't use cellular energy and only allow ions and molecules through the membrane that would normally pass through as a result of diffusion. However, as we will see later, there are carrier proteins that use cellular energy to carry substances against the flow created by diffusion.
Okay, so that video gave us an introduction into facilitated diffusion, and we've already talked about that regular simple diffusion. So again, simple diffusion is just what we've been talking about, where substances just go from high concentration to low concentration across the membrane itself. Facilitated diffusion is when we can move larger substances or substances that are unable to pass through the membrane, okay, across that membrane through some sort of carrier protein or a transmembrane protein. Okay, so here we have this large, channel protein or transmembrane protein, and we can. Same thing where we're moving the substance from high concentration to low concentration. So this is still passive transport, but again, we're using a protein involved here. So here we have our high concentration of some substance, and then it requires the protein to help it through. Okay, so that's facilitated transport. We have to have a facilitator to help it through. And excuse that spelling down there. It should be no energy expenditure. So no energy expenditure, just like with simple diffusion. This way, the only thing that's different is that we have a facilitator. We have to have a protein involved here to help carry that substance across the membrane. So again, this is where we have larger molecules or things that are polar, things that won't cross that membrane on its own. All right. So we're still moving from high concentration to low concentration. And then the final category of passive transport, which the video actually didn't go into, was osmosis. So in osmosis, this is the type of diffusion of water. So it has its own special category. And in osmosis, we're still thinking of water moving from high concentration to low concentration. However, because a lot of times there's different things dissolved in water, certain things can happen that are a little wonky here. So we have like a whole separate issue involved with osmosis. and that's when we get into things like tonicity and so forth. So with osmosis, we look at the solute concentration. So remember, solute is basically the substance that is dissolved within the solvent, and in this case, water is the solvent to form the solution. So when we're dealing with osmosis, we're looking at the concentration of the solute. All right. So let's take a look at an example here of osmosis. Here we have a semi-permeable membrane, and this particular semi-permeable membrane is only permeable to water. It is not permeable to whatever the solute is, this little purple ball. Okay, so you can see here that in this example, the purple ball is more concentrated on the right side of the beaker than on the left side of the beaker. So we have a higher concentration of purple balls on the right side than on the left side. But because of that, we have a higher concentration of purple balls, but that means we have a lower concentration of water. Okay, so it's more concentrated of purple balls, less concentration of water. All right, that means that on this side, we have lower concentration of purple balls, higher concentration of water. So based on that, remember, water moves from high concentration to low concentration. Since we have a higher concentration of water on this side of the beaker, water is going to want to move to this side to even out the concentration. So that's why you see the water moving to this side. And now we have an equal concentration. Now the purple balls are spread out. Okay, so now they're no longer concentrated on this side. We have an equal concentration of purple ball to water on each side. Okay, so I know it gets a little confusing, so don't worry, we have an Amoeba Sisters video. There we go.
When we were kids growing up in West Texas, our winters would be cold but rarely experienced snow. But we did have ice, which resulted in the roads being salted. As the salt mixes in and dissolves into water on the road, this can lead to a lower freezing point, which can help prevent the roads from icing over. And while this is great for making the roads more safe, it wasn't so great for the plants that lived right along the roadside. It often caused them to die. Now, winter can be hard for many plant species, but I'm talking about this salt affecting even some hardy plant life. This issue with salt in plants isn't limited to winter. During hurricanes near the coast, salty ocean water can be dumped in large quantities into the soil. This can eventually kill plants, including trees that had originally survived the hurricane. Why? Do plants just dislike salt that much? Well, it's actually related to a term called osmosis. When you are talking about osmosis, you are talking about the movement of water through a semi-permeable membrane, like a cell membrane. Water molecules are so small that they can travel through the cell membrane unassisted, or they can travel in larger quantities through protein channels like aquaporins. The movement of water molecules traveling across the cell membrane is passive transport, which means it does not require energy. In osmosis, water molecules travel from areas of a high concentration of water molecules to a low concentration of water molecules. But there's another way to think about water movement in osmosis. A low water concentration likely means there is a greater solute concentration. Solutes are substances like salt or sugar that can be dissolved within a solvent, like water. Water has the tendency to move to areas where there is a higher solute concentration, which would mean less water concentration. So if you want to easily figure out where the water will travel in osmosis, look to the side where there is a greater solute concentration. Unless we bring in another variable, like pressure, water will generally have a net movement to the area of higher solute concentration. So let's bring out a U-tube. U-tube, that's funny. There's a semi-permeable membrane in the middle of it. Let's assume that it is similar to a cell membrane and that water molecules can squeeze through it. The molecules are quite small, but salt can't. Right now there's just water in this U-tube. the water levels on side A and side B are equal, that doesn't mean that the water molecules aren't moving. Water molecules like to move, but the net movement across the two sides is 0. That means the overall change in direction of movement is 0. Now let's imagine on side B, you dump a huge amount of salt there. So which direction will the water initially move towards? A or B? Think about what we mentioned with osmosis. The answer is B. Side B has a higher solute concentration than side A. Water moves to areas of higher solute concentration, which is also the area of lower water concentration. You will also see the water level on side B rise as the water moves to that area. You can almost think of the water as trying to equalize the concentrations, diluting side B. Once equilibrium is reached, the net movement of water across the two sides will be 0, but remember that water still likes to move and movement still occurs. Now here's some vocabulary to add in here. We call side B hypertonic. That means higher solute concentration. But we can't just say something is hypertonic without comparing it to something else. We say side B is hypertonic to side A because it has a higher solute concentration than side A. In osmosis, water moves to the hypertonic side. We say side A is hypotonic. Hypo rhymes with low, which helps me remember that it's the low solute concentration when compared to side B. Let's get a little more real life now instead of just the YouTube. As you know, water is important for your body and many processes that occur in the body. When someone gets an IV in a hospital, it might look like the fluid in the IV is just pure water. But it is certainly not pure water. That would be a disaster because of osmosis. Let's explain. Let's say hypothetically pure water was in an IV. Now an IV tube typically runs through a vein so that you have access to your bloodstream. Really useful for running medication through. Blood actually consists of many different types of components, and red blood cells are a great example. So what do you think has a higher solute concentration? The hypothetical pure water in this IV tube, or the red blood cells? Well, cells are not empty vessels. They contain solutes. The pure water that hypothetically is running through this IV tube has no solutes. So where does the water go? It goes to the area of higher solute concentration, which in this case is inside the cells. The cells are hypertonic compared to the pure water in the IV tube because the cells have a greater solute concentration. The cells would swell and possibly burst. Exploding red blood cells are not good. If a person needs fluids, they typically will receive a solution that is isotonic to their blood plasma. Isotonic means equal concentration, so you won't have any swelling or shrinking red blood cells. Another example, let's talk about the aquarium. I've always wanted a saltwater fish tank ever since I was a little kid, but I've only had freshwater tanks so far. I did often question when I was a kid, why is it that a saltwater fish can't be in a freshwater tank? Well, let me explain one reason why this would be dangerous to the saltwater fish and how it relates to osmosis. First ask, where is the higher solute concentration? In the saltwater fish cells, or in the freshwater that the fish would be hypothetically placed in? Definitely in the saltwater fish cells. So where would the water go? It goes to the area where there's a higher solute concentration, the hypertonic side, so it goes into the cells of that poor saltwater fish. If not rescued, it could die. Now, one thing to clarify, saltwater fish and freshwater fish are not necessarily isotonic to their surroundings, but they have special adaptations that allow them to live in their environment and usually cannot make a major switch from a saltwater environment to a freshwater one. Now, not all fish have this problem. There are some fish that have this amazing adaptation to switch between fresh and salt water, and they have to deal with this osmosis problem. Salmon, for example. I think if I could pick to be a fish, I'd be a salmon. Osmosis explains how many kinds of plants get their water. Sure, many plants have roots, but how does the water get into the roots? When it rains, the soil becomes saturated with water. The root hair cells generally have a higher concentration of solutes within them than the solute concentration in the saturated soil. The water travels into the root hair cells as the root hair cells are hypertonic compared to the hypotonic soil. By the way, you may wonder, well, why don't those root hair cells burst with all the water that's going in them? That brings us to our next osmosis topic, and why plant cell walls are amazing. So let's bring in another variable that can influence osmosis: pressure potential. This is when it's very useful to understand how one can calculate water potential. Water potential considers both solute potential and pressure potential. In osmosis, water travels to areas of lower water potential. So the formula is water potential is equal to the pressure potential plus the solute potential. Adding solute actually causes the solute potential to have a negative value and the overall water potential to lower. Water will travel to areas of lower water potential, but exerting pressure can raise the pressure potential, a positive value, therefore raising the total water potential. So let's give a quick example. In the popular water potential in potato cores lab, all kinds of neat variations of this lab procedure exist online, you can calculate the water potential in potato cores using the water potential formula. When a potato core is first put into distilled water, that's pure water, the potato core cells start to gain water. You'd expect that the water is moving towards the higher solute concentration. Thanks to their higher solute concentration, they have a lower solute potential. That means a lower total water potential than the surroundings, and water travels to areas of lower water potential. But over time, as the potato core cells gain water, the water that has entered exerts pressure against the plant cell walls from inside the plant cells, therefore raising the overall water potential in the potato core cells. We want to point out that this turgor pressure that results in plant cells, thanks to osmosis and plant cell walls, is critical for overall plant structure and the ability of plants to grow upright and not wilt. Turgor pressure is definitely something to explore. In summary, where would living organisms be without osmosis? After all, it involves movement of one of our very valuable resources, water. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so that was a good introduction into this whole thing of osmosis and tonicity and hypotonic versus hypertonic versus isotonic. So let's get into it. So first off, let's do some definitions, make sure we have the terminology down. So tonicity. Tonicity, we're referring to the solute. So remember, we're talking about in osmosis, we're looking at the concentration of water, but that's in relationship to the concentration of the solute. So tonicity is the amount of solute in the solution. So if we are looking at the amount of tonicity, the measure of tonicity of the solution, we're basically looking at that compared to the amount of water. So that adds up to the osmolarity. Okay, so the total amount of solutes dissolved in a specific amount of solution. So that's the osmolarity. So when we're looking at basically the concentration of water to solute, we end up with three types of solutions. We can either have a hypotonic solution, a hypertonic solution, or an isotonic solution. And the Amoeba Sisters video gave us some kind of, ways to help us remember. So think of hypo as low solute. Okay, so low solute solutions. So this is where we have a lower concentration of solutes outside the cell than inside the cell. Okay, so because, again, we're talking about the solution that the cell is in. Okay, so that we have a cell, we place that cell inside a hypotonic solution. So what happens to the cell when it's in a hypotonic solution? Now there's less solute outside the cell than inside the cell. So there's more water outside the cell than inside the cell. Okay, so because there's more water outside the cell than inside the cell, the water is going to want to move into the cell. Because remember, water moves from high concentration to low concentration. So here again is an example. So we place that cell into a beaker of hypotonic solution. So now we have low solute outside of the cell. We have high water. So if we have more water outside the cell than inside the cell, the water is going to move from high concentration to low concentration. So it's going to move into the cell. As it's moving into the cell, our cells, our human cells, remember, don't have cell walls. And so it's going to end up gaining so much water that it's going to burst. Okay, so you can see it here is bursting. These red blood cells are bursting, or what's called lysing. Okay, so lysis will take place, or bursting. All right, so hypotonic solutions, we have hypo, low solute, high water concentration. All right. Hyper is the opposite. So think high solute, low water. So higher concentration of solute outside of the cell. So if we have a lot of solute outside the cell, that means we have low amount of water outside the cell. So there's going to be more water inside the cell than outside. So water is going to leave the cell. As it leaves the cell starts to shrivel up. Okay, so that's what we call crenation. So the cell shrivels or crenates. So this is what happens to our little red blood cells if we place them in a hypertonic solutions. They start to shrivel or crenate because they lost all their water to the surroundings. So hypertonic, think high solute, low water. So that causes the cell to shrivel. And then finally, iso, think equal. Okay, so equal concentrations of solute inside the cell as outside the cell. And so in this case, since it's equal concentrations of solute, it'll be equal concentrations of water. That doesn't mean that the water stops moving. It will still move, but it'll just move at an equal concentration. So that means there's no net gain or loss of water. Okay, so this means we have happy cells. Okay, so isotonic would be what the cell wants to be in for our animal cells. So you can see these are our red blood cells. So this is what it wants to be in. You see the nice concave shape of our red blood cells to carry the oxygen. This is what it wants to be. So isotonic, think equal concentration. So this is our tonicity with our animal cells. So hypo, low solute, high water. Hyper, high solute, low water. Iso, equal solute, equal water. All right, so this, we've looked at the consequences of these solutions to our animal cells. Let's see what happens when we place plant cells in these solutions. There are some differences because plant cells, they have cell walls. And because they have cell walls, they act a little differently, okay? So when we place a plant in a hypotonic solution, remember hypo is low solute, high water. That's when the water is going to want to enter the cell. The cell in a plant cell has a cell wall surrounding it. And because it has a cell wall, the cell can only expand so much. And that's going to prevent it from bursting or from lysis taking place. Okay? So a hypotonic solution, that cell will gain water, and here you can see the water is filling its central vacuole, but eventually there's going to be so much pressure on that cell membrane against the cell wall that it's going to prevent the cell from absorbing any more water. Okay, so that's what we call turgor pressure. So turgor pressure is that pressure that is placed on the cell membrane against that cell wall, and it'll prevent that cell from bursting. Okay, so water simply can't move any more water into the cell. Okay, So that's a way that it prevents the plant cell from bursting. Okay, animal cells, we don't have a cell wall, so our cells can burst if it's placed in a hypotonic solution. Plant cells, they just simply don't take up any more water because their cell walls prevent them from doing so. They are protected, right? In a hypertonic solution, remember hypertonic, high solute, low water, so water will leave the cell. So as water leaves the cell, you can kind of see what happens. The cell membrane kind of shrinks away from the cell wall. But the cell wall is still there. It still provides structure to the plant, okay? Just the cell membrane kind of wilts away. And that's what we call plasmolysis. Okay, so think plasma as plasma membrane, lysis kind of think, well, it's not really bursting, but it's kind of shrinking away from the cell membrane. So it's not fully crenating, but it is shrinking away, all right, as the water leaves the cell. And this causes our plants, like, you know, what happens when you forget to water your plant? It wilts, right? But it doesn't completely like disintegrate unless you've forgotten to water it for a long time and then just completely dies. But it will still maintain its shape for a while until you actually kill it, right? Okay, it'll just wilt away a little bit. All right. So those are some new terms that you just have to kind of remember are associated with the plant cells that the animal cells don't have. Okay, so animal cells, we don't have cell walls, and so we don't have trigger pressure. Our cells just burst when we're in hypotonic solutions. We don't have plasmolysis, okay, when we're in a hypertonic solution. Our cell walls our cells walls don't maintain the shape of our cells here because we don't have cell walls. Our cells just shrivel up. Okay. All right. So that is so we've talked about passive transport. We've talked about the 3 types of diffusion: simple diffusion, facilitated diffusion, and now osmosis. So now we're going to finish off this video with some review questions. So water moves via osmosis, A, from an area with a high concentration of other solutes to a lower one, or is it B, from an area with a low concentration of water to one of a higher concentration, or is it C, throughout the cytoplasm, or is it D, from an area with a low concentration of solutes to an area with a higher one? So water moves via osmosis. So how does water move? If you chose D, I would agree with you. So remember D, we have a lower concentration of solutes, which means we have a higher concentration of water. So low concentration of solutes, high concentration of water, to an area of high solute, low water. Okay, so we just have to kind of reword this. So D is the correct answer. It's not A, because if we had a high concentration of solutes, that means we have low water. And water is not going to move from low water to high water. That would be against its concentration gradient. Okay, so D is the correct answer. Here's another question for you. On a hot summer day, you forgot to water your plants. They are now all wilted and drooping. What happened to the water inside the plant cells? Is it A, the water moved into the plants, increasing the trigger pressure? B, the amount of water stayed the same inside the plant cells, causing no change in trigger pressure? Or C, the water moved out of the cells, causing plasmolysis? If you chose C, I would agree with you. So remember the water, if it's moving, if it's losing water, that means that the water has moved out of the cell, causing plasmolysis. Okay, and that is a wilted plant. Okay, if water has moved into the plant, it would look a lot happier than that. So that would not be A or B. So C is the correct answer. Here's another review question. The principal force driving movement in diffusion is it A, temperature, B, particle size, C, concentration gradient, or D, membrane surface area? So what's the principal force driving movement in diffusion? If you said concentration gradient, I would agree with you. Okay, so it's the concentration gradient that causes molecules to move from high concentration to low concentration that leads to diffusion. Alright, so that's it for our first video on the working cell. So now that you understand passive transport, you should be able to explain why and how it occurs. You should also understand the process of osmosis and diffusion, as well as define tonicity and describe its relevance to passive transport. Okay? So in the next video, we're going to get into active transport. So remember, passive transport requires no energy. So now we're going to get into active transport, which requires energy. Okay? And it requires energy because we're going to go in the opposite direction. We're going to move molecules against their concentration gradient. We're going to go from low concentration to high concentration against their concentration gradient. Okay, so we'll see that in the next video.